I'd like to welcome Professor Vijay Tiwari to the floor. Vijay is the Executive Scientific Advisor at 𝐁𝐢𝐨𝐬𝐚𝐫𝐤𝐞𝐫𝐬. He has an internationally visible record in genomics, epigenetics, and computational biology, with many high-impact papers in Nature, Nature Genetics, Cancer Cell, Cell Stem Cell, Nature Communications, you name it, basically. And he's here to speak on the topic of how genomics can be used to drive healthy living. Vijay, thank you so much for joining us today. The floor is yours.
Thanks. Yeah, thanks, Franco, for this introduction. And I'm glad to be in this meeting. I'm already learning a lot. So as Franco said, I'm an academic lead at the Queen's University in UK. At the same time, I'm a former director of biomarker discovery at a Skylight in Switzerland and currently serve as an executive advisor. So I will present some flavors of both of these, but basically related to genomics and healthy living. So I drew this slide a few minutes before the meeting to draw your attention that what we call healthy is very tricky because it has multi-parameters. It can have some clinical definitions. For example, you have gone through a checkup and everything looks fine. Externally, you feel good. You don't show any symptoms of mental disorders. You may have no genetic predisposition. And of course, your lifestyle, how good you exercise, how well you sleep, do you have good work-life balance. So to define a good and healthy lifestyle is a multifactorial process, and it's very difficult to come to a solid conclusion. And when we say we know healthy, only then we start to call what is a deviation from healthy. So there is serious need of developing a baseline pattern for a healthy person. But what is becoming clear that a sensitive and early detection of this deviation from healthy is key to a healthy living and longevity. And I will give you a few examples, including history of genomics, how it has started to influence our lives, and of course, aging. We know we have 3 million base pairs, around 22,000 coding genes. Among us, we have quite some similarities, and we are 99.6% similar, and we are 99% identical to chimpanzee genome. Yet we look very different, as you might notice. It all began in 90s when the Human Genome Project was launched. The project went very well, and we ended up having the first version of Human Genome completed. And you'll be surprised that some parts were still missing. It actually was completed last month. But this led to a lot of new discoveries and new changes. For example, the moment we had a genetic code sequenced, we started looking for which variations within our gene associate with distinct disorders. So first, genome-wide association studies were published. Then came the ENCODE project that aimed to measure the functional regulatory regions in the genome, including DNA methylation, histone modifications, non-coding RNA, and subsequently mapping genome of cancers. And this obviously led to a lot of other subsequent studies, including development of knockout mutations, first full sequencing of the Neanderthal genome, and so on and so forth. So basically from sequencing to genome-wide studies to basically understanding how genes affect our lifestyles. And this is a chart I draw with Nature published in 2011, how they saw genomics will influence us. So the 90s to 2003 was the Human Genome Project. Then we saw more development in understanding the biology of the genomes. From 11 to 20, we have seen a lot of studies on understanding the biology of disease. And now projection is that from 20s to 30s, we would expect to see its clinical impact. And this is the time when we start to harvest all this data derived from genomics and try to make an impact on human lives. So this is where we are at the moment, and this is the right time to make use of it. This is also hampered, had been facilitated by a number of sequencing instruments that have hit the scene in the last decade. And the advantage has been that the sequencing costs have gone down significantly. There's a decrease of time as well per genome sequencing. And we are discovering more and more genetic variants that associate with different disease risks. And in the end, from that same genomics information, We are able to discover copy number variation, discover mutations, understand DNA protein interactions, transcription profiles, metagenomics, metabolomics, RNA, splicing, you name it. So this has really revolutionized the way we understand about how the normal physiology of our body and how these things go wrong in diseases. I'll give you a few examples how this has started to facilitate early diagnostics and thereby healthy living. So one of the first noticeable project was 𝐁𝐸𝐬𝐷 project, where there was comprehensive genome sequencing to search for individual genetic variants. There they focused on exomes. That was 1.5% of total genome. It's cost effective when you want to sequence many people. And the first large scale whole genome sequencing project was called 𝐶𝐬𝐶𝐬𝐿 Genome project. That was most robust, accurate test compared to exome sequencing. It replaced exome sequencing because the cost decreased, and obviously it could detect much more things beyond exome. So what we have understood that we have a lot of single nucleotide variants that stand among us. Many of these are also multi-nucleotide variants that include indels, copy number variations, inversions, translocations, and aneuploidy. And this led to development of a number of consortium that have been running last 10 to 15 years to establish genetic variants of specific disorders. I give you a few examples. One is Alzheimer's disease consortium, was type 2 diabetes, then psychiatric consortium. And they have already started to show which gene variations might predispose us for these disorders. Another noticeable example is genetic risk factors for type 2 diabetes. A number of nations around the globe have launched a lot of genome sequencing projects a few years ago. And once compiled, this is an unprecedented level of information that would be of use. And obviously, this then went to commercialization. A number of companies, they started offering personal genetic testing. Among these, the most noticeable one was 23andMe, that all of us probably would have heard about. So far, they have sold more than 10 million kits. 80% of their customers consent to research. They have collected 2 billion plus data of phenotypic points. And they take advantage of DNA sequencing. They sell as kits. They collect phenotypic data through self-report surveys about wellness and behavior, serious conditions, rare disease, for example. And then they try to relate this to the genetic composition. One of the first case of genomics-based preventive medicine was highlighted by Angelina Jolie that led to a lot of awareness among individuals. She did her genome sequencing, and she found she harbors mutation in BRCA1 that increases the risk of breast cancer by 87% and ovarian cancer by 50%. She decided to undergo double mastectomy. And basically, that way she reduced her risk of actually developing this problem. What we start to see basically is human body is a source of big data. At the moment, we collect a lot of MRIs, we collect mammograms, we are starting to collect CT scans, we have X-rays. And so at this moment, on an average, every hospital has 665 terabytes of data. And on top of this, now next generation sequencing data sets will also become a common practice among clinicians. So that will add terabytes of data. So data management and integration is becoming critical. This is another example of how big genome sequencing projects have facilitated targeted therapy and giving longer lifespan to patients. For example, this TCG consortia that aim to sequence almost all tumor types, as you can 33 different tumor types, 10 rare cancer, 11,000 patients in the beginning. And using this profiling, they could find that certain types of mutations predispose us or make us chemoresistant to certain kinds of therapies. For example, in this case, alterations in the gene EGFR, they found they respond to treatment tyrosine kinase inhibitors for lung cancer. So if you do not have this alteration, this inhibitor is useless. You waste your time in your life. And there were several such similar examples in this case. So knowing your genetic variation allows one to tailor the treatment in the clinic and thereby helping you support live a better life. Similar was the case on ALL, where it was found that the patients with this genetic profile were originally incurable, while these were treatment responsive. Based on this genomics mutation and gene expression profiling, they could identify new drugs that could actually treat these originally incurable patients. So that also allows to find newer drug targets. In terms of therapy development, this has led to a lot of progress in new drug development as well as identifying new targets. And this has also given rise to a new field called pharmacogenomics. And this is because we have gradually learned that 99% of people have genetic variants that influence how we process in response to medication. The same drug might not work equally effectively in both of us. So thereby, that genetic predisposition to differential response must be understood. So medications, for example, may be processed too fast and removed from the body before taking effect, or processed too slowly, thereby building up in the body, causing side effects. And this is determined by metabolism, which in turn is influenced by gene expression programs or genetic component composition. Individuals may be prone to side effects or unintended drug reactions. So this is why this field has a lot of promise for future, where based on genomics profile, we could actually know and learn which drugs I may respond better to and which one should not. And this is already developing very rapidly. So this is a number of publications, a number of databases are being developed that aim to actually predict individual response to drugs. They also want to rationalize drug development and design. They also want to repurpose drugs, some drugs that existed, but we did not know where they could be more effective. And they also want to use this information to better select individuals for clinical trials and also find uses for drugs that failed earlier trials because we did not have the genetic information. So there's a lot of data-driven prediction of drug effect. For example, this is one program called Proctor. It's a tool that directly predicts the likelihood of toxicity of clinical trials because it integrates chemical properties and tissue-specific target gene expressions using random forest model to predict whether the drug is likely to fail clinical trials for toxicity reasons. Obviously, this has also led to learnings about the epigenetic control of gene expression. We know that things that change within the cell are, in terms of gene expression, are controlled by a variety of histone modifications, DNA methylation, chromatin cross of transcription factors, which in turn are influenced by extracellular environment. So nothing happens in isolation. The environment here induces cell signaling pathways that in turn influence chromatin, that in turn influences gene expression, and then obviously affects the cell phenotype. So there's a lot of effort in targeting chromatin because tumor cells not only are activated by genetic and epigenetic alterations, but also they misguide these processes to escape chemotherapy and host immune surveillance. So these have become new targets for therapy and intervention. The Epigenomics project that was launched a few years ago aimed at mapping epigenomic state of every human tissue type, and it has been completed with great success. So my lab at Queen's, we try to integrate epigenetics, genomics, systems biology, computational biology to understand predisposition to neurological disorders, find tailored therapeutic approaches as well as in cancers. In the end, to enable early detection and promote a healthy living and enhance longevity. One of the major problems in the field has been a late detection of many diseases. For example, 4 out of 10 will develop some kind of cancer on this planet. We have 15 million new patients each year. And out of these, half of them are diagnosed late. And that comes because of the current diagnostic methods. What we do, we take bulk tissue and we look for markers or changes in cell types. We cannot detect these orange or bad cells in the beginning because if it's 1 or 2 cells wrong here, the bulk methods would not be able to detect because we have a pooled population. So 𝐁𝐢𝐨𝐬𝐚 company in Switzerland aims to do this analysis at a single cell resolution to be able to detect these abnormal cells at the very early stages of a problem. When we say single cell data, they exist in many forms. So at the moment, we have possibility of doing cytometry analysis. We do a lot of single cell RNA sequencing, so profile transcriptome at the single cell level. You could do T cell, B cell receptor sequencing. And now multi-omics profiling is becoming very common. For example, we do a lot of combined ATAC-seq and transcriptome profiling. So from the same cell, you can get gene expression and chromatin accessibility. From the same cell, you can get gene expression and DNA methylomics profile. So this has revolutionized to basically diagnose early and as well as understand mechanisms of the onset of these diseases. So 𝐁𝐈 has developed an artificial intelligence-based algorithm we call 𝐁𝐢𝐨𝐬𝐚 that has ability to analyze these single cell data sets to a very sensitive and accurate levels, thereby allowing to identify biomarkers that characterize a sick population. And this has facilitated early diagnostics and patient stratification, for example, for a therapy. This has been very successful. As you could see here, so far we have had 6 projects and biomarkers have been identified in 3 of these projects. We have patented many of these and currently going to develop an IVD prototype to bring it to the market. So these single cell based diagnostics tools have shown power to provide biomarkers to detect disease early and hereby preventing or treating the disease better. All in all, the need of the hour actually is to manage these big genomics data sets in a more professional way and allow its much wider uses. And in this sense, given the scale of the data, deep learning or AI is becoming very relevant at this moment. So overall, so far, we have been looking in a very narrow dimensions with very limited physiological parameters to understand or promote healthy living and or delay problems in our life. And this is basically the risks, diagnostics, and treatment. If we know our risks, if we can diagnose early, and if we can treat in the right way, we can live a longer, healthy life. But there has been a lot of darkness around it, given the previous gaps in the knowledge. But with the onset of genome sequences, with the transcriptome maps, with the epigenome maps, with the pharmacogenomics and genome-wide association studies, this is becoming very easy. And this is the right time to make use of this, to have this multimodal integrated pathway to understand how to live a better, longer, healthy lives. So genomics will have huge impact on diagnostics and characterization and treatment of diseases. It will promote preventive medicine, and is doing already, hence promoting healthy living and longevity. It will allow us to stratify patients using whole genome sequencing, gene expression, or epigenomic profiling, thereby paving the way for personalized medicine. Not only that, it will predict the toxicity of new promising therapeutic targets. And there is need of development of new experimental and computation analysis to generate and integrate multi-layer of genomics data. And obviously, artificial intelligence has a lot of promises for it. And as I mentioned, good management of these big genomics data sets is key for the most effective use of all these data around to enable a healthier and longer life to individuals on this planet. With that, I thank you for the opportunity, Franco. I hope I'm in time.
You are. Thank you so much, Professor Tiwari. Very insightful. For any of our viewers who have specific questions on 𝐁𝐈 and Professor Tiwari's work, please remember to submit them to the Q&A box for the panel discussion. Make sure to stick around for that panel discussion because we'll be bringing all of today's speakers together to delve into these various topics in a more dynamic and deeper manner. And also, for those viewers who might be interested in hearing more about intersections between genetics and genomics, make sure to stick around for day 2 tomorrow of our 2-day conference. which will focus on sector spotlight and diversity of practical applications. We do have a few talks that touch upon that as well tomorrow.